All right, got my chat window up. Okay, that's better. All right, I think we're getting people back.
Yeah, it's just that people are starting to figure. It's gonna take some time for people to figure out. Okay, I got kicked out of a meeting. Now I'm trying to get back in. So.
All right, so everybody who's connected right now, let's see how many I have. All right, I have 26. Hopefully we get the rest quickly. I'm going to send out some commands in chat that you'll need to run. Usually I would share these on the board. Oh, and I need to actually pull up the other slides too. All right, is everybody seeing a slide that says collaboratory AP at the top? Okay, cool. All right, so obviously ignore the top part. Nobody should be in the Boyer Hall classroom right now, but what you're going to do is you're going to log in to the Hoffman cluster. So log into the Hoffman cluster through your terminal program like SSH or whatever you have. And you're going to run the following commands. If anybody runs into issues running these commands, let me know. And once you manage to get these commands run, you should, so after you run QRSH, your terminal is going to hang for a minute or two while it gets you an interactive session. Then the next command is going to be the CP command. That'll be copying data over. That's also going to hang for a minute or two while it copies. You'll go into your Scratch folder and you'll open up the tar archive. So once people are done opening up the tar archive, just respond in chat saying done so that I get an idea when it finishes for people. Depending on how heavily the Hoffman cluster is being used right now, sometimes it takes just a couple minutes. Sometimes it takes like 5 or 10. Actually, while you're doing that, I'm going to get myself a drink really quick. I will be right back. Yeah, you'll probably see something that says no H data set. That's perfectly fine. That's just kind of a default message that's telling you to set a specific amount of memory. All right. Has anybody's untar finished yet? Okay, you'll know your untar finishes when you get back your command prompt. So if you have it, so if you ran the tar-xvf command and you have a command prompt back, then it finished. All right, so if you're still either running this or waiting for it to finish, just hit the raise hand button so I can see how many people are still waiting. Okay, I see quite a few people still pinging on that one. So okay, I'll give it a few more minutes. All right, and we'll be right back because I have my dog demanding to enter the room. So I'll be back in just a moment. All right, so for Soyun Kim, it looks like the command you typed, you're missing the $ scratch at the end of it. Soyun Kim, can you respond if that helped? Okay, perfect. So I think just about everybody's gotten it done. All right, I think just about everybody has this done, so I'm going to move on. I've left the commands in chat in case anybody else needs it. All right, this is a part I've had to add to the class because we've had some issues with it in the past. But I call this part, this is why we can't have nice things. And an issue that you're going to run into, you might run into it in this class depending on what the ads are doing, but you will definitely run into it if you try and get bioinformatics software online, is fake download links. So at least at one point, if you went to the CyberDuck page, you would see this link right here. And it turns out that this link is not really a download link. It'll download something to your computer. I don't know what it is, but you don't want it. But it won't be CyberDuck. The good link is actually down here under the duck. This is a bad link. That link is typically going to be some form of malware. All right, some other things that I've seen pop up during class, people will get these fake maintenance alerts saying your computer is infected. Your web browser never knows if your computer is infected. So if your web browser is saying your computer has a computer virus on it, there's usually a pretty good chance that it's actually just some website trying to get you to download something that is probably computer virus itself. Occasionally, they're even smart enough to know that you're on a Mac. And so that's it for that part. So just keep an eye out because everybody probably knows by now the internet is full of scammers. All right, is everybody seeing my other set of slides now? So you should be seeing a slide that looks like a command prompt. Okay, awesome. All right, so what we're going to do today is we are going to start looking over our FASTQ files. So I'm going to show you guys how to do quality control on a FASTQ file. And there's actually a really good app for doing quality control on FASTQ files, and that app is called FastQC. Has anybody out there used FastQC before? Okay, a couple people. Yeah, FastQC is a really good app, and I'm going to show you how to run it on the cluster. Does anybody know where to download FastQC from? Okay, so I'm guessing a lot of people don't know where to download FastQC from off the top of your head. Can anybody guess how to figure out where to download FastQC from? Yep, Google. So the three secrets to solving just about any sequencing and bioinformatics problem, at least initially. Use Google, read error messages, and read the manual. And those 3 steps will solve probably 90+% of your problems. All right, so everybody Google FastQC. And when you Google FastQC, this should come up pretty quickly. So you'll see this is a FastQC homepage. And what we're going to want to do is we're going to want to download it. We're not going to be running FastQC on our own computers, though. So in order to download it to the cluster, we know we're running it in a Linux environment because that's what's on the cluster. You're going to right-click on this link, specifically the one that says Windows and Linux zip file, and you're going to copy the link address. All right, I have somebody saying unpacking the tar file is taking forever. It does take a few minutes. Are you seeing a bunch of text go across your screen? Okay, if you're seeing a bunch of text going across your screen, it's definitely working. It can take a few minutes to unpack. The tar file is a little bit bigger than it needs to be. There's actually, so the tar file includes some extra FASTQ files so that you have some fresh files to play around with later if you want to get a little bit more practice. All right, so once you've copied that link, you're going to go onto you'll go into your terminal, which should be in an interactive session, and you're going to CD into your WS2 folder. So it'll be CD, and then Workshop 2, and then CD WS2, and that'll get you into this WS2 folder. Yeah, essay index can take a minute or two. Although for some reason it seems like the cluster is running a little bit more slowly, because usually this goes a little bit faster. All right, so once you're in your WS2 folder, you're going to use the command wget, and wget is how your program gets some resource off the internet. So it'll be wget and then space, and then you'll paste in that web address we just got, and double check to make sure the web address ends with .zip. If the web address ends with .dmg, you accidentally copied the Mac version, so you'll need to go back and replace it. Don't try to copy what's on here. Number one, it's long and complicated and you'll probably make a typo. And number two, this is a version that's actually a little bit older. Yes, CD Workshop 2 and then CD WS2. All right, so this is a good opportunity to use tab autocomplete. All right, and I see Shuya Wang is having some trouble. Oh, you can't type them both on the same line. All right, so I'm going to keep going. Yeah, if it worked with just CDWS2, that means you're already in the Workshop 2 folder. All right, so once you're in the WS2 folder, that's when you're going to type in the wget command. So you'll wget that link that we just copied from the FastQC page. Okay, yeah, sorry, I had it reversed. It would be WS2 and then Workshop 2 data. All right, for Che Chen, can you paste the address that you put into the wget command? Did you remember to put a space between wget and the address? All right, try doing it one more time just to see if it works. Is that command not working for anybody, or is that command just not working for one person? Did anybody get the command to work? Okay, just try the command again. It most likely is either there was something weird with the way it got copied in there, or alternatively, there may have been an issue because everybody was trying to download it once or something. So give it another couple tries. It seems like the site is valid, so it should be there. All right, so once you've copied this into your WS2 folder, and copying it once you've gotten the wget command working should only take a couple of seconds. Type ls, and you should see that you have a new file in there called fastqc, and then some version number .zip. And since we're mostly going to be using this fastqc with our workshop 2 data, let's actually move the zip file into workshop 2 _ data. And then once you've done that, hit ls, and you should see that the fastqc zip file is gone. And then we'll cd into workshop 2 _ data. And that's actually where we're going to be working for the remainder of the class. So copy it down a level into workshop 2 _ data, and then go into that folder. And then the next command we're going to do is we're going to unzip the fastqc program. So we'll do unzip fastqc and then whatever it is. I highly recommend getting used to using tab autocomplete. So you should just be able to type fastqc or probably even just FA and hit tab, and it should autocomplete to the file name. Using tab autocomplete doesn't make you lazy, makes you smart, because it prevents you from making a lot of typos. And as soon as you unzip it, you're going to see a whole bunch of stuff go across your terminal, just like when you ran the untar command. And that's just the zip file opening up. So I'll give everybody a minute or two on that. So the command to unzip is actually just unzip. So a pretty standard pattern in Linux is a lot of the commands actually kind of are, some of the commands are intuitive, like unzip. And the nice thing is most of the time you'll give a command and then you'll just give a file name to work on, or you'll give some options for how to do it. So in the case of unzipping, the command is just unzip and then the file name. All right, is there anybody who isn't able to get the unzip command to work? All right, I'll go back for a moment to the previous commands. All right, I think everybody should have this command going and I'll get moving on. All right, so we should still be in the workshop 2_data folder. And typically, you're gonna see me do this a lot. This is a pretty common pattern, is once I do something, I'll frequently hit ls. So just run ls and you should see that you have a new folder created called fastqc. So let's cd into that fastqc folder. Remember, capitalization matters and it's gonna start with a capital F probably. So cd into that fastqc and type ls to kind of have a look around and see what's in there. Once you look around in there, you'll see that there's a file called fastqc with nothing after it. So typically in Linux, if you have a file that doesn't have anything after it, that's probably an executable file. So we'll try and run it by typing /fastqc, which is basically telling it just run. You have to give it sort of a more of a path to actually run a file. So if you do /fastqc, that'll tell it to try and run fastqc. It's going to give you a permission denied error. And that's because in Linux, there's a few different kinds of permissions files need. So if you run ls-l, and this is also an important command to remember, ls-l will give you kind of the long form file list. You'll be able to see that, you'll be able to see a few different things about the file. So you'll see the sort of general information column. You'll see who the owner is of the file, what group it belongs to. This is the file size in blocks, which isn't really human interpretable. Information on when the file was created, and then the file name. What we're really interested in is right here, which is the file information. So for example, the first column, you'll see some of them have Ds. All the files with D in this column are actually directories, so you can CD into them. The next columns, and you'll see 9 of them, go in groups of 3. And these are the actual permissions. So the permissions go as groups of 3. The first 3 are for the user or the owner. The second group of 3 are for the owner's group. The third group of 3 are for everyone on the system. And within a group of 3, there's also R, W, and X, and that's read, write, and execute. So for example, for this first file, I have permission to read and write it. I don't have permission to execute it. But since I'm the owner, I can give myself permission to execute. So in the case of this fastqc file, I can read it, I can write to it, but I don't have permission to execute it. That's easy enough to fix though. The command to fix a permission is chmod. So it's going to be chmod. And there's a few different ways to do this, but the simplest one is just chmod and then space and then plus X and then another space and then fastqc. So what the command is saying is change permissions, add execution to fastqc. And then we'll run /fastqc again. Oh yes, if you haven't downloaded the slides, it's a really good idea to do this. I don't know what video quality you're getting on your end, but it's going to be much better video quality if you get the slides. Do not, however, try to copy and paste things from the slides into the terminal. That's going to end badly for you. Okay, that's a new error message. You got that when you tried to run /fastqc? And this was after you gave permission to run it, or? Huh. Okay, I'm not quite sure what that is. I haven't seen that error before. Okay, that's an error I haven't seen before. I'm wondering if it's just a new error message from it. It might be because it's trying to, I don't think it's because you need a gig of RAM, because I've been actually running this class for a few years now on a 1 gig RAM, 1 core system, basically what everybody gets when they default QRSH. And this command has worked previous classes. So unless either the cluster changed or fastqc changed significantly, Hoffman changed policies. Okay, apparently Hoffman changed their policies. Okay, I guess I need to change my slides then. I didn't know that they had a policy change recently. All right, so in that case, I'll need everybody to use the following commands. So what we're going to do is we're going to we'll terminate our current interactive sessions. And you can terminate an interactive session by just typing logout. And then I believe to get 4 gig node, which should be more than enough to run everything we're doing, you just do QRSH-L space QRSH space -L space H data equals 4G. And that should get you a 4 gig node, I believe. All right, so it's going to take a minute or two to get that done. Basically, once you've put in for your interactive session, since that's going to take a minute or two, let's all use that opportunity to take kind of a bio break, stretch, whatever you need. So it's 10:06 right now, so we'll pick up again. Yeah, we'll pick up again at let's say 10:15. And if anybody has trouble getting a session going, let me know, and I will see if I can help you out.
Am I going to make a quick announcement?
Okay.
Yeah. So everybody, I want to apologize about the complications earlier today. So when I had to start a new meeting, I meant is I was going to restart this meeting. It's more complicated for me to just create a new meeting and send out passwords and register, and everybody who's registered. So when that happens, it resets the. When I restart this meeting, it resets it so that Dr. Weinstein can come back into the meeting. So I apologize if anybody got we're waiting out for 15 minutes and so sorry for the. I'll try to keep that in mind in the future. Okay.
All right, the recordings are up on my Google Drive, and I just posted a link to them. And that actually will give you access for those taking Workshop 18. You'll also get access to those because those are in the same folder.
Hey, Michael, is it cool if I could download those and put it on our website as well?
Yeah, go for it.
All right,
you should be able to download from the link.
All right, cool. Thank you.
Mm-hmm. All right, for those asking, you don't have to rerun the unzip and tar again. That's data already saved to the hard drive. All right, and Anand, the error that you're showing is one that I expect you to get, where it says headless exception. That's actually an error that I was expecting people to get, so that's fine. All right, for the people who are saying they got an error saying failed to map segment from shared object, cannot allocate memory. I'm not sure exactly how that error is happening. Did you put in for a new interactive session and you use -L, and you did -L and then space H_data equals 4G? All right, we'll see what it does with the next command. It's possible that might be an error that it's getting because it's trying to launch through graphical interface, which is usually not what we do. All right, so it's 10:17 now, so everybody should be back. All right, the next command we're going to do is since FastQC didn't want to run without what they call a headless exception, all we really need to do is we can actually skip this command because that one was one where I was typing and I made a mistake. So we need to remember it's /fastqc and then space and then /human001_pe1.fastq. So when you try and run that command, you should get fastqc kind of counting off how much of the file is done. All right, for So-Young Kim, you should already be in the fastqc folder. If you're not in the fastqc folder, it should be go to your Scratch. Oh, that's right. Okay, so you're going to do CD, and then it should be CD and then $Scratch. All right, I just typed in the command you should be able to use. All right, was anybody getting any weird errors when you tried to run fastqc, giving it a file name to work on? All right, so where it says skipping /human001 for Ellen O'Connor, you should have a slash between the ... and the H. So for Ellen O'Connor, try applying that solution and let me know if it works. Okay, cool. All right, once this is done, do a Google search to try and find CyberDuck. And everybody should go and download a copy of CyberDuck. Yes, if you have a different SCP, if you have a different SCP program, like FileZilla, or there's like a couple bajillion of them for Mac, or there's WinSCP. If you have your own, feel free to use it. If you have your own, I also suspect you might have probably configured it already to connect to the Hoffman cluster. So everybody's free to use their own. I'm going to show people how to set up a connection. CyberDuck you're going to put on your local computer. So CyberDuck is how you move files between your computer and the Hoffman cluster. All right. Once you have CyberDuck downloaded, depending on whether you're on a Mac or a PC, you'll see a slightly different user interface, but the ideas will still be the same here. All right, everybody should be seeing a screen share of my CyberDuck now. So I'm on a Windows machine, so my interface will be like this, but either way, you're gonna wanna go to Bookmarks. And if you're not seeing something that looks like this with a bunch of bookmarks in it, then you're going to wanna go to Bookmark and then toggle bookmarks, which in my case makes them go away because I already had them, but toggling bookmarks again brings them back. You'll see somewhere on your screen, if you're on Windows, it'll look probably a lot like this, an option to add a new bookmark, which is gonna be a plus. We'll click on that. And it'll give you an empty bookmark. So we're going to edit the bookmark. And you'll see a window that looks just like this. So we're going to use SFTP. That should automatically change the port number to 22. And we'll leave it at that. We're going to change the nickname to Hoffman 2 home. And in my case, I'm calling it Hoffman 2 home class because I already have a Hoffman 2 home link and that'll get confusing. The server is going to be dtn2.hoffman2.ucla.edu. The username is going to be your username. And in my case, it already has a password saved, so it's going to keep using that same password. And the path we're going to set, if you don't see this option down here to set a path, because you may not have it clicked, then just click on more options. It'll give you the option to set a path. So the path is going to be /u/home/ the first letter of your username. So don't copy me on this part, otherwise you'll try and connect to my folder. /your username. So I'm going to leave this up here for a minute. And again, remember, don't copy this portion from mine because that's for my username. It's going to be the first letter of your username / your username. Is there anybody who wasn't able to get this done who needed it? Yeah, it's going to be /u/home/ the first letter of your username on the cluster / your username. Okay, and then once that's done, all you actually have to do is hit the X to exit out of here. So I'm going to hit this and you'll see me lose my screen share for a second. All right, the next trick we're going to do is probably for a lot of you, this is going to be the only, I also lost my chat window. There we go. So for a lot of you, this is going to, this link we just made will be the only link that you have. So you're going to right click on it and you have the option to duplicate the bookmark. So we're actually going to duplicate this bookmark and it's going to pop up another window, which I will start sharing now. All right, so we're going to change the name from Hoffman 2 home to Hoffman 2 scratch. And again, I just have class there because I already have these links and I don't want to get rid of them. We're going to keep just about everything the same. With one exception. We're going to change the word home here between those slashes to scratch. So I'll give everybody a minute or two to do that. And I'll give everybody a minute to get that sorted out. And I'll answer some questions in the meantime because I see a few questions coming in. So if you're already connected with your username to the Hoffman cluster on FileZilla, you should be good there. Just navigate to your Scratch folder. All right, so if you're in FileZilla, I don't have FileZilla on this computer, so I can't walk you through it exactly. But essentially what you're going to want to do is navigate up to the /u folder, and then navigate back down to the /scratch / the first letter of your username / your username. So the Scratch file system's on a completely different drive. All right, and I think by this point, everybody should have that link set up. So essentially what you just did was you made 2 connections to the Hoffman cluster. One of them goes directly into your home folder. One of them goes directly into your Scratch folder. We're going to actually connect to the Scratch one right now, because Scratch is where we're doing all of our work. So I'm going to close down this link creating window, and I'm going to lose my share for just a moment. All right, so everybody should be back on my slides. All right, so once you get onto the Hoffman cluster, navigate from, you should be connecting directly to your Scratch folder. Navigate down into workshop, navigate into WS2 and then workshop2_data, where we've been doing all of our work. And you should be able to see the file we just created. And the file we just created ends in .html. So it'll probably have an icon next to it that looks like Google Chrome or Firefox or whatever browser you're using. If it has an icon next to it that looks like Internet Explorer, get a better browser. All right, for Marcella Haro, who just asked, where is the Scratch folder? Did you connect with CyberDuck? Okay, you have FileZilla. Or is FileZilla a different program, or is that just a typo? Okay. So you should be able to navigate to that folder. It's so you should be able to navigate to that folder with the following link. You'll just need to replace the stuff I have in brackets. So it's /u/scratch/ the first letter of your username / your username. All right, so once you get into that folder, copy the HTML file that we just created using FastQ. Just copy it onto your desktop and then double click it. It should automatically open up in your internet browser. And I'll give everybody a minute or two to get that done. Okay, the file we're going to be copying. Let me actually advance a slide. I'm going to advance one slide, not two. There we go. All right, so we've created a file called human001_pe1_fastqc.html. So this is a file we're going to be copying. It's the only one that should end with .html. So All right. Sorry for the delay. I'm just trying to help a few people who got a bit behind. All right. Just give me one more minute, trying to help a student who got a little bit behind. I'm trying to help get them caught up. Yeah. For those asking how to for the one asking how to copy the HTML file to your desktop, from CyberDuck, you just drag and drop it over. You can just drag and drop it from CyberDuck into your desktop, and it'll automatically copy. Okay, so going on from this, you should be able to open up the file in your you should be able to open up the file in your web browser. You'll see something that looks like this. And the important stuff to note is you'll have basic statistics on your file. So it'll tell you, for example, how many reads. It'll tell you how many sequences were flagged as being poor quality. Since we have no sequences flagged as poor quality, the file's already probably been filtered. We have our sequence length. We have our %GC content. We also have our encoding. So this is actually telling us what kind of quality score encoding we have. You'll also see along here that we have our summary, and we see that we have a big red X next to per base sequence quality. So this big red X tells us there's something wrong with per base sequence quality. So options here, we can either give up or we can do a little bit of cleaning. So where are we right now in our pipeline? We have to clean up our raw reads. So this is just a paper that is kind of nice if you're new to sequencing. It'll help you, it can kind of help you understand some of the issues you're running into with bad quality sequence and that sort of thing. And this will help us understand why we're getting adapters in our sequencing data. So cleaning sequence frequently involves a combination of trimming away garbage from the ends of sequence, as well as occasionally throwing away sequences that are just poor quality. So one of the things we want to get rid of is what's called adapter contamination. So when I showed earlier how Illumina sequencing works, you have adapters on either end to help it to start off sequencing and also to bind it to the flow cell and provide barcodes for multiplexing and all kinds of other good stuff. But it turns out that if your insert is a little bit short, you can read all the way through the insert and start reading 3' adapter. 3' adapter is not part of the genome. So if you start reading 3' adapter, that's actually artificial sequence. That's sequence that you shouldn't really be trying to align and trying to analyze because it's completely bogus. So we want to get rid of that. We also want to get rid of low quality sequence. So low quality sequence, for anybody who's done Sanger sequence, it's just the same general idea. The further into a DNA sequence you read, the lower your quality tends to get. So by the end of longer Illumina reads, you'll tend to notice there's some drop off in quality. Sometimes there's a big drop off in quality and you really want to get rid of that stuff because it's garbage. So when it comes to trimming, there's a few good options. There's what's called hard trimming. And hard trimming is cutting off the reads after a fixed number of bases. So a lot of times we'll be cutting off the reads after about 50 bases. Or, you know, for a 100 base read, you may say, well, the quality starts to rapidly decline after base 70, so I'm just going to cut off at base 70. It's good for certain things, so it's especially good if you're doing RNA-seq, and you're doing RNA-seq just to do differential gene expression. Hard trimming is actually a very good option there. Hard trimming is very fast, and it takes minimal computational resource. There's also what I call smart trimming, and smart trimming is quality score aware. Oh, the other advantage of hard trimming is it'll end with all of your reads being the same length. So when you get back your raw reads, they're typically all the same length because Illumina goes in cycles. But if you do hard trimming, you're just trimming them all back to the same length. If you do what I call smart trimming, which is quality score aware. Okay, and I'm going to pause for 1 second. I have a question. So do we need adapter sequence to know where the read came from, though? If you mean for the purpose of multiplexing and demultiplexing, usually by the time you're reaching the cleaning stage, you've already demultiplexed. And beyond that, the adapter information is actually frequently stored in the read header. Yeah, as far as the actual barcode sequence goes, the barcode sequence should not be in your FASTQ file as part of the read, because the barcode sequence is also artificial sequence. People call it either artifact, artificial sequence, or technical sequence, but whatever you want to call it, the barcode is part of that. So you don't want barcode to go into your actual DNA sequence read that's being claimed to be from the insert, because that's not part of the genome that you're trying to analyze. So again, if you do smart trimming is what we're actually going to do is with a program called Sickle. And Sickle is reading a certain size window in each read. And from that window, it's saying once the quality score in the window has dropped below a certain threshold, we're going to actually cut off the read there. So it'll look for where the read starts to decline in quality beyond a certain point, and it'll just cut the read off. So that was nice because that'll maximize the amount of read you get to keep. Your reads will wind up being different lengths, but you know, it's not a big problem. So Talking again about quality scores, and just reminding you, the quality scores run on scales and they're encoded using ASCII. This is that rule of thumb I mentioned yesterday. So a Fred quality score of 10 is a 1 in 10 chance of error, 90% confidence. 20 is 99% confidence, 30 is 99.9, and 40 is 99.99% confident. Typically for reads, you won't go above 40. The machines just won't, even if they're more confident than 1 in 10,000, they'll still just call it 1 in 10,000 confidence. And what we see here are two different charts, and you'll notice that for both of them, on the y-axis, we have values going 0 to 38. Well, typically quality scores get around to 40. Along the x-axis, we have positions in the read, and we can already see here that as we go further in the read, we have declining quality. So the way we fix this is just by doing some trim. How did the run go? There's a few other metrics that we're interested in. So sequence duplication level. We typically want this to be peaking above 1. We don't want to have a lot of sequence duplication. You can get sequence duplication from two different ways. One of them is because during the library prep, you usually have a PCR step. You can get sequence duplicates that way, where something just gets PCR too many times and it winds up getting overrepresented in the reads. It's especially bad if you do a library prep method that requires a lot of PCR. So just as an example, the person who ran this class before me, she was doing genomes on the, she was actually doing genomes on the deadliest animal on planet Earth. Anybody know what the deadliest animal is on planet Earth for humans? Well, besides other humans? All right, I got mosquitoes and I got hippos. Given the choice, I'd actually rather face off between, I'd rather face off with a mosquito than either a hippo or some spider. But yes, the actual deadliest creature on planet Earth for humans, besides other humans, is the mosquito, especially like the Anopheles mosquito, which basically mosquitoes are just nasty and they transmit all sorts of parasites. So in her case, she was studying, I think she was studying the Aedes aegypti mosquito, which is another one of the big parasite transmitters. And it turns out mosquitoes don't have a lot of material to them. They're really tiny, they don't have a lot of DNA. And if you do a total DNA extraction from the entire mosquito, you still don't get enough DNA for a really good library prep. Go figure. So in her case, her library preps had to actually involve a lot of extra PCR, and she was having horrible problems. Sometimes about 30% or more of her library was actually PCR duplicate that she couldn't use. So PCR duplication can be a big issue if you have kind of a low input library. Another thing that can happen is occasionally when you do that colony formation process I talked about, although this is less of an issue now, I think Illumina's done a few tricks to make this less of an issue. You can occasionally get a colony that kind of split in two, and the Illumina machine would interpret it as two separate colonies. Again, they've gotten much better at not doing this, so optical duplicates I haven't seen be a huge problem lately. Plus, you can also identify them by the fact that they're the same sequence right next to each other. Also in RNA-seq, because you have a lot of non-random stuff, RNA-seq can have some stuff that's called as duplicates that may not be duplicates. All right, we're also going to look at frequencies across frequencies of different bases across each cycle. Yes, and Marco is actually mentioning that, yeah, the pattern flow cells. So previously, when I showed you that video, the flow cell looked like it just had a lawn of sequences for the adapters to stick to. But Illumina's now doing a thing where the flow cells basically have these kind of micro dimples in them. It's basically a flow cell with millions of microscopic wells in it. And because of that, the sequence can only bind to a specific microwell, and you wind up not having issues so much with optical duplicates. But I believe that feature only came into play on the Hi-Seq 3000 and after. So if somebody's using an older machine, and I still see the occasional Hi-Seq 2500 around campus, optical duplicates are something that could potentially happen there. And just FYI, Marco totally beats me at Illumina trivia. So anyway, questions of how did your run go? You're also going to want to look at base frequencies across different cycles. And Marco says not sure, so I'm saying challenge accepted. So you want to look at base frequencies across different cycles. And what you're going to see is typically with a lot of reads, you're going to notice there's a little bit of noise at the beginning, which is because you have some non-random things happening. Like you have non-random, you'll have non-random ligation of primers to sequences. So sometimes the primers might preferentially ligate at high GC regions or things like that. So you can have some non-randomness in the beginning, but what should happen is very quickly it evens out. And you see pretty much even across the reads. If you start seeing big changes, like you see a bunch of unexpected spikes, that's a big cause for concern because that means something may have gone wrong with your sequencing run. Like for example, if you notice that somewhere around cycle 20, there was a big jump in Gs, that means there may have been some kind of a fault with the machine. And again, you'll see non-randomness at the beginning of your reads, but very quickly after that, you'll start seeing it go even. This is common in RNA-seq again, because RNA-seq has a much higher degree of non-randomness in how it's done. For example, grabbing sequences by the poly A tails will tend to cause that kind of thing to happen. If you see a bunch of really weird noise, that means you might have adapter contamination. It might mean some other things too, but if you see a lot of if this plot looks really noisy, it's generally a cause for concern. All right, so a program called Scythe is what we're going to use for trimming off adapters. Scythe is what's called quality score aware. So all Scythe is going to do is it's going to look at the 3' end of your read. It's going to compare it to an expected adapter sequence. And in this case, we can see it lines up pretty darn well. We have two mismatches, this A to C and this T to G. But since Scythe is quality score aware, Scythe is actually going to say the base quality score here at these two mismatches is pretty low. So these are low confidence base calls. So what it's going to say is, okay, we have a very high confidence calls that match. The two mismatches are low confidence base calls. This is more likely than not an adapter. For those who like statistics, it's almost certainly using a Bayesian method to determine this. So that can kind of give you an idea of what it's doing. If not, then it's just determining a statistical likelihood that this is adapter sequence versus it is not. In this case, we see there's a bunch of mismatches relative to the adapter. And a lot of these mismatches are really confident base calls. So what it's going to say here is, I don't see the adapter. And in fact, this final AG could actually be adapter, but it's probably going to say that it's hard to call. Trimming with sickle. Sickle is going to move a sliding window across the reads. And by the way, sliding window. So quickly, I have a question from the audience. Do you have to input your adapter seek? Yes, you have to tell. You have to explicitly tell Scythe what your adapters are, and we'll be doing just that in a moment. I'll show you how to do that. So this is a bioinformatic algorithm. It's a fairly simple one called sliding window, where you're just sliding a window of a certain size across your sequence. So in this case, it's showing 8. I think in reality, sickle uses about 15, but it's sliding a window across. And with each iteration of the program, it's just moving the window over by 1. It's averaging the quality scores for these bases. And once the quality score drops below a certain value, so for example, Fred's score of 20, where we'd be saying the average base is 99% confident. Once the average base in the window goes below 99% confidence, SYCL is going to say cut off the read here. Has anybody ever tried to align sequences that are less than, say, 25 or 30 bases long? Anybody out there in class ever tried that? All right, I'm not seeing much response, so I'm guessing not a lot of people have tried that. That's a good thing. Trying to align very short sequence to the human genome is typically a waste of time. They'll align to a lot of different places. They're not very confident, and it's just generally a bad idea. So as you can imagine with SYCL, where you're trimming off the reads after a certain quality score, occasionally you're going to trim the read, and you're going to have maybe 15 bases left. You don't want to try and align 15 bases to the genome. That's not a good idea. That's going to just waste time. So occasionally with SYCL, it'll trim the bases down too much, and it has to throw out the entire read. So occasionally SYCL will get rid, well, at some rate, SYCL is going to get rid of reads from your sequencing. But there's a little bit of a trap here to be careful of. So the way paired end reads tend to work is you have paired end file 1, paired end file 2, and most programs are going to take one read from each file at the same time. And what's going to happen is read 1 from paired end 1 and read 1 from paired end 2 are going to be consumed by the program. It's going to do whatever it does. Then it'll pull the next set of reads from the program, and it should be read 2 and read 2. Pretty much every program on Earth expects this. So if after your trimming, you've gotten rid of read 1, read 1 and read 4, then you're now going to go read 2, read 3, read 5, read 6, read 7, and they're going to be completely mismatched. So one of two things is going to happen here. Less likely the programs will still handle this, and you're going to look like you have genomic rearrangements all over the place because you now have read 2 looking like it was connected to read 1, read 3 with read 2, read 5 with read 3, and so on. It'll look like you have a tumor or something with a lot of crazy chromosomal translocations. But more likely, you're going to have a scenario where the program compares the read identifiers and immediately realizes that read 2 and read 1 were not from the same read, and it's going to throw you an error message telling you to go fix your out-of-sync files. Either one of those is going to bring you to a stop. So the way SYCL deals with this is SYCL just goes under logic that if I throw out paired end 1, read 1, paired end 2, read 1 is no longer paired. It's now a singleton, and SYCL will pass those off to a singletons file. All right, so these are kind of some generic command line formation for these. All right, and a couple tricks running on the command line. You have to read the manual. Reading the manual is very important. Be careful with spelling. The command line doesn't typically tolerate spelling or even capitalization mistakes. So you'll frequently get error messages. Also remember, software is written by different people. So what you're going to find is that the way you interact with different pieces of software is going to vary. There's a few things that you'll find just about every programmer does. Like for example, command line options are pretty much always handled the same way because everybody uses the same libraries to handle command line inputs. But the way programs behave can be very different. So if you're ever wondering why two pieces of software, it seems like you interact with them completely differently, it's because they were written by very different people. All right, so both Scythe and SYCL are programs that are written in either C or C++, and they have to be compiled. So that's turning the human readable source code into machine readable binary code. So if we're in our workshop 2 data folder, we'll do ls, and we'll see that we have a folder called software. So we'll go into software in ls, and we'll see that we have 2 zip folders, scythe and sickle. And we'll just use one command. We'll use one command line to unzip them both. So we know the unzip command from before. Does anybody know what this semicolon does? Anybody out there know what the semicolon does on a command line? All right, it's not pipeline. Pipeline is actually a different character. What the semicolon, the semicolon basically says treat everything after the semicolon as a new line. So it lets you do two separate commands on one line. And so the 2 commands we're going to do here: unzip scythe.zip, unzip sickle.zip. And I think the reason I actually even did this here was I just wanted to get up and get a glass of water. So as soon as you have this command entered, it takes about a minute or 2 to get these guys unzipped. So once you've typed in that command and it's going, feel free to get up and get yourself a glass of water, do whatever you have to do. And then we'll pick up in, let's see, it's 11:07. I'll pick up in about 3 more minutes, but that should give everybody time to get these guys unzipped. All right, I think everybody should have gotten them unzipped by now. So you'll see a whole bunch of stuff go across your terminal just like before. And then when you ls, you should see that you have 2 new files created. So these 2 files are actually folders. So we'll go into the folders now. So let's do cd and then scythe master. So again, a good place to use tab autocomplete. And if you do ls, you'll see a couple of key features here. The main ones I want people to start noticing: you have a file called makefile, and you have a folder called src, which is source code. that means that this is a program that was written in C or C++ and it needs to be compiled. And the cool thing is with Makefile, that actually has all the instructions for your compiler to automatically compile it for you. And the only thing you really have to do to cause it to start is you just type make. So if in ScytheMaster you just type make, it'll do the compiling for you. And then we'll cd .. /SickleMaster. So we're just going to jump directly over to our SickleMaster folder, which is our other program. We'll ls in there, we'll realize that it has a Makefile as well and an src folder. And we'll just type make there, and that'll cause that folder compile too. All right. Is there anybody who's getting errors trying to compile the programs? All right, I think everybody should have compiled by now. Is there anybody who hasn't been able to get it to compile yet? All right, just do a quick show of hands if you've gotten both Sickle and Scythe compiled. Okay, looks like everybody got them both compiled. Cool, because we're about to test to make sure they're working. All right, so we're going to do cd and then .. /.. So we're going to go back into our main workshop2_data folder. And we're going to start working on the command lines for running Sickle and Scythe. So the first command we'll do is /software/sickle-master/sickle. Again, I highly recommend using tab autocomplete for these when you can. So you'll just tab a couple times. You'll be able to tab and then autocomplete each of these words once you have enough of it. So we'll try and first run Sickle, and when you try and run it, you're going to get a splash screen that looks something like this. So a lot of software, if it requires parameters or files to work on, and Sickle obviously will, Sickle is going to instead just display you kind of a mini manual showing you how to work it. So again, just kind of our generic command. So if you get that splash, it tells you that SYCL is actually up and running. But the first one we're going to run is scythe. So we'll actually just do our proper scythe command to get going. And the command we're going to do for that is going to be very similar to what we did for SYCL. It's going to be /software/scythe-master/scythe. And then make sure you copy this part very carefully. And remember that these characters at the beginning of the file name are numbers because we're just numbering the files to show which step we're on. So the first file we're going to create is 1_sci_human001_pe1.fastq. And I'm going to assume everybody has the slides available in front of them because I'm actually going to show you what the next step we do is. So for the next step, and I'm going to warn people now about, people call this steamrollering files or clobbering files, or occasionally I think the technical term is overwriting files, which you can do unintentionally. So I'm actually going to jump back really quickly. If you notice the command we ran here, it's just PE1 in this file and PE1 in this file. The next command we're going to run is just switching the PE1s to PE2s, but again, make sure you switch both of them. If you don't switch both of them over, what will happen is you can either overwrite the file you just created, or you can accidentally run PE1 twice. So if you forget to change the file we're operating on, you'll run PE1 twice and then write it to PE2. If you forget to change the file we're writing to, you'll overwrite the original file with PE2 data. So I'll let everybody run both PE1 and PE2 in their own time, and I'll give a few minutes for those to happen. I have a question. What's the slide number for the first scythe command? All right, for the first scythe command, you're going to probably be looking at slide 130. So I will let everybody get those going. All right. Is there anybody who feels like they're hopelessly stuck? If you feel like you're hopelessly stuck, send me send something in chat. Okay, and I got a couple people raising their hands. So send me something in chat letting me know what issues you're having. All right. I think a lot of people should have been able to get this. Should have been able to get this run already. So I'm going to keep on moving. So for running SYCL, it's a little bit more of a complex operation because it has to work on both paired ends at the same time. So SYCL we only run once, and it handles both files. And keep in mind the files we're going to be giving it are the ones we just created with side. So the command we're going to run for this is /software/SYCL-master/SYCL. So this command here, PE is telling it we're running in paired end mode. And then we have to give it a -F. So that's our forward reads. Our -R, that's our reverse reads. Our -T, and that's saying it's Sanger type quality score encoding. -O, and that's going to be our first output file. So that's going to be our paired end 1 output file. -P, that's going to be our paired end 2 output file. And -S, that's going to be our singletons output file. So what we're giving it is essentially what it needs. Is we're giving a command to run SYCL. We're telling it it's running in paired end mode. We're going to give it a forward read file, which is the one we just made with Scythe. We're going to give it a reverse read file, which is going to be the reverse file we just made with Scythe. We have to tell it the quality score encoding, which is Sanger. And then we're giving it 3 output files: a forward output file, a reverse output file, and an output file for the singletons that lost their paired end mates. All right. This should take a couple minutes to run. Can people put up hands if they've gotten this process completed so far? All right, I see a lot of hands going up, so I think this part's done. Awesome. Okay, so I can get us on to the next step. All right, so once you finish up that, you can ls, and you should be able to see that we have some new files we've created, including the ones starting with 2. So let's rerun fastqc. Let's just rerun fastqc on our sickle created file. So it's going to be 2_sick_human001_p1.fastq. So we'll run sickle on, or we'll run fastqc on this file, and this is going to reevaluate how our file looks after it's clean. So once fastqc is done, go into CyberDuck, and you should be able to copy over the file over to your desktop, and you should be able to open up the new file. All right, when you run the command and you just get the splash screen, there's a very good chance that you probably have the command wrong. So what I want you to do is copy the command you used to try and run it into the chat window, and I'll see if I can troubleshoot you from there. Once you run the, okay, so for Jennifer, once you run the sickle command, you're going to, once you run the sickle command, you're going to run fastqc on the parent N1 output you generated, and that's going to help us reevaluate how we change the quality of our reads. All right, and for Anand, who sent me a private message trying to look at an error message, I'm going to ask you to resend that as a chat to everyone, because if you're having that problem, other people might be having the same problem too, and I prefer to be able to help everyone. Okay, there we go. All right, and also don't forget to include the error message with it. All right, so you're getting a message saying no such file or directory. So that would mean that you're either trying to run from the wrong folder, or you didn't do a make on your sickle program. All right, so for everyone who's gotten it, just copy that that most recent HTML file to your desktop, open it up in your browser. And when you look over the basic report now, you should see that the sequence quality has improved. So as soon as you've gotten the FastQC report open on your browser, and you see that we've gotten better sequence qualities, I think I'm going to end it there for today, and I'm going to stick around and help people who got behind on the material and see if I can help them get caught up for tomorrow. So as soon as you've gotten the most recent FastQC report opened, you're free to go, and I will stick around for a few minutes to answer questions and help anybody who's having difficulties. Okay, so if you're getting a permission denied error, that's going to mean that you didn't run the chmod command. All right, for those who are behind, I just shared the Google Drive address again. You should be able to follow along the slides from there. That should have all the commands you need. All right. I will see everyone tomorrow. If you still run into questions, feel free to send me an email and I'll see if I can help you out. But I think we've hit the end of our meeting time.